Welcome to Bacteria and how it impacts our everyday life. So in this demonstration, students will gain the understanding of what bacteria is, where it can be found, how it is grown, and a common lab procedure to see its growth, along with how it's transmitted and the difference between pathogenic and non-pathogenic bacteria. So first, we're going to go over some background, and we're going to go over the bacteria structure. So first you have the capsule, which is the part of the bacteria that allows them to attach to surfaces and get nutrients, but also helps in protecting them. You then have the cell wall, which is another thing that provides protection, but it also helps in giving it its shape. You have the plasma membrane that surrounds the cytoplasm and helps with the flow of substances inside and outside the bacteria. You have the pilus, and these are going to be tiny hair-like structures that allow bacteria to attach to other bacteria. Some of them also help in attaching to surfaces. You have the flagellum, which helps with cellular motion. You have the ribosomes, which are going to help in protein production. You have the plasmid, and that's going to be where genes are found. And in bacteria, there is circular DNA instead of linear DNA. You then have the nucleoid region, and that's the part of the cytoplasm that has a single bacterial DNA molecule. And now we're going to go over the main shapes of bacteria. And so the first one is going to be Kochia bacteria, and these typically are described as round, oval, spherical. Then you have bacilli bacteria, and these are going to be rod-shaped cells. And then you have spirilla bacteria, and those can be described as twisty. And those are three main shape categories of bacteria. So there's also two types of bacteria. You have pathogenic and non-pathogenic. And so pathogenic bacteria is the bacteria that often causes sickness, infections, and in extreme cases, death. And then you have non-pathogenic bacteria, and these are bacteria that cause no harm for the most part, and they're mainly there to kind of like support, provide benefit to the environment that they're in. So now during this part of the video, you will stop it, and I'm going to ask you 3 questions. So first, where can bacteria be found? Second, how can we see it? And third, what is an agar plate? And these questions are going to be answered like 1, 2, 3 in the order that they're listed above. And so just pause this part and take a minute to respond, and then unpause it. And so now we're going to grab our lab coats because we're going to be growing our own bacteria colonies. And so this is going to be done using an agar dish. And since I don't have one right now with me, I'm just going to demonstrate with my hands and just like a regular object. So what you're going to do first is that you want to sterilize your station and wash your hands so that no unwanted bacteria ends up there. And then you're going to grab your dish. So we're going to have a dish. And then you're just going to open it, and you're going to grab a little swab, and you're just going to swab any location you feel like it. As long as no hazardous bacteria is growing there. So avoid places maybe like your kitchen sink and the bathroom. And then you're just going to gently swab over the section, making sure that it's really spread out. And once you're done with that, you're going to close the dish, discard of the swab, and then you're just going to let it sit in room temperature, and over time you should see that bacteria colony grow. And then the last step is just making sure that you clean up your environment. And so now we're going to play a little game And in this part of the game, you're going to rank the following items. So you have a keyboard, your hands, the bottom of your shoe, the bottom of your backpack, the back of a phone, your hands with sanitizer, your keys, and then the front of your phone. And then you're going to pause it here, and either in groups or by yourself, however it works best, you're going to list these items from what you feel would be the cleanest to the dirtiest. And then at the end of the time needed, I would probably say maybe like 5 minutes. You can go back and have a discussion about what everyone thought was the cleanest versus the dirtiest item and see who got the closest to the actual results. And that is going to be in these next slides. And so you will see that the dirtiest or the cleanest object was your hands with hand sanitizer. And then the second was the front of your phone. Your keys, and then it goes the front of your phone as third. The fourth is going to be the bottom of the backpack. The second is going to be the bottom of your shoe. The sixth one is going to be your hands. The seventh one is going to be the back of your phone. And then the dirtiest one is going to be the keyboard. And then also here you would pause it and go over if these are the results you expected, why or why not. And also quickly, you can also mention the fun fact that because keys are metal, bacteria has a harder time to survive on it, and that's why it's ranked number 2 in terms of how clean it is. After this, you're just going to wrap up with some important takeaways. So having them know that bacteria can be found anywhere, it comes in many forms, that it can be either pathogenic or non-pathogenic, that they're very tiny, and so we need to find different ways to be able to see them. Also go over how bacteria is spread, so through air, inhalation, droplet, water, food, etc. And that's why it's important to do things such as blocking a sneeze or a cough in order to not spread bacteria, and then knowing that good habits result in less bacteria, pathogenic bacteria to be specific. And so that in order to not get sick from bacteria, it is important to do things such as wash your hands, be more clean with surfaces, avoid touching your mouth, eyes, nose, especially if you've been touching different surfaces and your hands aren't clean, and then just cleaning up surfaces after they're done. And then finally, just wrap up with a little affirmation. I put here that remember that you are all stars and you shine so bright. You are capable of so much, and each one of you are going to do so many amazing things. So keep dreaming, learning, and always believing. Thank you for watching this video. I hope it is useful.